Hello, and welcome back to our discussion of the immune system. Today's topic is the adaptive immune system. This is part 1. And as always, here are your learning objectives with the corresponding sections in your textbook. Okay, so as always, we're going to start off with a glimpse in history, and this is a good one. Remember when we discussed gram-positive bacteria, we talked about Corynebacterium diphtheriae. So this is the bacteria that causes diphtheria. And so there's sort of a progression to this, and I kind of want to stress that it's never one person that comes up with a great discovery. It's always built on other work. And so let's just kind of follow how we got to the point where there's no longer diphtheria for the most part in the United States. So let's look at Frederick Loeffler. He was the one that found the club-shaped bacterium, Corynebacterium diphtheriae. And so he suggested that this bacteria made a poison, which we know is a toxin, that spread through the blood. Then it was Emil Roux and Alexander Yersin that sort of built on his research, and they extracted the poison and they injected it into guinea pigs. I know, they're so cute. It was usually fatal, but there were some guinea pigs that survived. And so what they did was then they took they injected that toxin into those guinea pigs that actually survived and realized that they didn't get it again. And so they suggested that there was something in their blood, these recovered guinea pigs, that protected them. And so that was called antitoxin. So then what they did was pretty smart. They took the serum, so serum is blood plasma minus the blood clotting proteins from these recovered guinea pigs, and they mixed it with a toxin. So they mixed their antibodies, so from their serum, and they mixed it with a toxin. We looked at this picture before. And so what they were injecting was this. And so when antibodies bind to a toxin, that neutralizes the toxin so it's not harmful. So they injected that into guinea pigs, and they realized that the guinea pigs remained well. And so this is the basis of antibiotic therapy. So this is what's being done when there's a disease. They did this with Ebola. So they took people that recovered from Ebola, pulled their serum, and someone that was currently in the throes of the disease, they would then inject those antibodies to those people to give them protection. They're also, this is one of those treatments for SARS-CoV-2 as well. And so antibiotic therapy. And so they were just sort of building upon this research. And then it was in Berlin in 1891 on Christmas, don't you just love a good Christmas story, that the antitoxin was first given to a child. And then that child recovered. So prior to this, diphtheria, before treatment, diphtheria was fatal in over 50% of cases. That's meaning half the children that got it died. And just kind of reminding you what it was, it causes that pseudomembranous pharyngitis, that then this can break off and block respiratory tract. This also causes a cytokine storm. Even with treatment, by the way, so this is more current, this is off the CDC, with treatment 5 to 10% fatality rate because of that cytokine storm. And if the child is under the age of 5, that's 20%. And so we came up with a Toxoid Vaccine was developed starting in the 1930s. And so in the United States, it's pretty much been eradicated, although it is making a slight resurgence due to the lack of vaccination. So yes, those anti-vaxxers. So strangely enough, so they did all this research. They knew about this sort of antibodies, and they came up with antitoxins, and then they also came up with treatment and the vaccine. But it really wasn't until 70 years later, basically, roughly, after the discovery of this sort of antitoxin, that they could actually crystallize and look at the structure of antibodies. And so that was also a Nobel Prize winning discovery. So 1972. So it was just sort of like a nice little glimpse of the progression to get to where we are today. And so I hope you enjoyed that. All right, so you've seen this slide before. So the last chapter was over innate immunity. And so our immune system, once again, we are basically one big nutrient-rich petri dish. And this is kind of really evident when you see what happens to a dead body when the immune system is no longer functioning. It takes hours before you start to get decomposition. And so the interior of the body is maintained relatively sterile due to our immune system. So we talked about the innate immune system, and so today we're going to talk about the adaptive immune system. So this is the one that it adapts, it develops over a lifetime, and so it's going to get better. This part of the immune system is slow if your immune system has never seen that antigen, so that's naive, and so we'll talk exactly about what that is. It is going to fight specifically to a particular antigen, and we'll see just how specific that is. It's going to respond by making antibodies, which are going to neutralize and target things for destruction. And because the next time you're exposed, because it has memory, it's going to have a stronger response to re-exposure. And so just kind of reminding you what we're discussing today and comparing it back to innate immunity. All right, so let's of course always start off with a great video just to get you all ready.
The adaptive immune system provides a more targeted defense. Dendritic cells are like intelligence specialists. They collect pieces of the invader at the site of the battle. Then they travel to the nearest lymph node, triggering a cascade of events. Inside the lymph node, the dendritic cell presents pieces of the invader to T cells, the immune system's communication specialist. In turn, T cells activate B cells, training them to form antibodies, the molecular weapons that target the invader specifically. The immune system now mounts its ultimate defense. B cells release millions of antibodies, each antibody's shape tailored to identify and neutralize the enemy. The immune system deploys these tailored weapons until the infection is wiped out, ready to respond even faster the next time.
Okay, so from that video, hopefully you got the characteristics of the adaptive immune system. I'm going to tell you that this is the most effective means to eliminate an invader. We cannot live off of our innate immune system alone. It is carried out exclusively by lymphocytes. This branch of the immune system has molecular specificity, so it is distinctly targeted to a particular virus, a particular bacterium, even to just a particular piece of the virus or bacterium. There is memory generated upon each exposure, so the body will have a stronger response the next time it's exposed. And so this is what vaccination relies upon. Also, this part of the adaptive immune system has to learn tolerance. It has to distinguish between our healthy self and something that is dangerous. And so we're going to look a little more closely at all of these things. All right, so the adaptive immune response, there's two basic strategies for eliminating foreign material. There's what's called humoral immunity, and that word humor refers to fluids or all the gushy parts of your body. It's an old-timey word. And then there's cell-mediated immunity. So that's implying this is brought about by cell contact. So your goals are to compare and contrast the characteristics, so let's go ahead and take a look. Your humoral immunity is going to eliminate extracellular antigens, so things like bacteria, toxins from those bacteria, those virion particles, so viruses in the bloodstream and tissue fluids, basically anywhere but not inside a cell. It's going to involve B cells, so those B lymphocytes. The effector B cells, so this word effector means this is the population of B cells that's actually going to bring about an effect to eliminate that invader. So those effector B cells are called plasma cells. They make antibodies, those Y-shaped proteins we just saw on that YouTube. You can drink antibodies. They're really not toxic on their own, but they are very powerful in the fact that they're going to neutralize pathogens and target them for destruction. Also, this humoral immunity, these B cells are going to leave behind memory cells that are going to remain for a quicker, stronger response the next time that person is exposed to that same antigen. And so these memory cells are going to bring about humoral immunity. Pop over here to cell-mediated immunity. So in contrast, cell-mediated immunity eliminates intracellular antigens, so things inside of the cells. So virus-infected cells or cancer cells. It involves T cells, not B cells, and so T cells are also called T lymphocytes. There's two populations as opposed to one on the humoral side. There's two effector T cells. One population is called the T helper cells, and these are the communicators that video was just talking about. They're going to activate, they're going to direct and assist the other cells of the adaptive immune system. Cytotoxic T cells, that name is telling you what they're doing. Cyto means cell, toxic means death or damage. Cytotoxic T cells are the effector cells that can actually kill the virus-infected cells or the tumor cells by cell-to-cell contact. Cell-mediated immunity, just like humoral immunity, leaves behind memory cells that are going to remain for that secondary exposure at any time that person is exposed to that same antigen throughout the course of their lifetime. So in both branches, it's going to be the memory cells that lead to immunity. So this is just a pictorial representation of the video we just watched, and also basically kind of what we just walked through. The innate immunity and the adaptive immunity, they work together. They're interconnected, but we have to break them up somehow to study them. So I don't want you to get the idea that they're acting independently. And so this picture is showing you that. Dendritic cells, we know, are phagocytic cells of the innate immunity. They are going to basically phagocytize and process antigens and then bring them to the lymphocytes for presentation. And so this presenting of the antigen, and here we're looking at a naive helper T cell and a naive cytotoxic T cell. Another name for them other than being T lymphocytes for the T helper cells is CD4 cells, and another name for the naive or the cytotoxic T cells is CD8 cells, and I'll show you exactly how they got their name in just a second. But it's the dendritic cell's job to present this antigen. Once we have antigen presentation, there's a secondary step that's called activation. And so there has to be presentation of the antigen, and there has to be activation, sort of a checks and balances thing, before we activate the adaptive immune system. And so what's part of that activation, this cell right here, this helper T cell, is necessary for both the humoral and the cell-mediated immunity. And so we've got activation. So this dendritic cell can activate the CD4 cell. The CD4 cell is going to talk to this macrophage. And remember, our communication between cells is always going to be those cytokines. So they're going to talk together. CD4 cell is going to get activated. This naive cytotoxic T cell is going to meet its antigen that's being presented by the dendritic cell. It's going to get activated. So now we have some activated cells. What's going to happen next is what's called proliferation and differentiation. Proliferation is a fancy word for dividing, and so these populations are going to divide and clone and copy themselves. Remember, the job of the T helper cells is to communicate and to activate and to stimulate and coordinate the immune response. So it's going to release cytokines that's going to then activate this naive B cell. So this naive B cell has to meet its antigen. It's going to get activated, and then it too goes through proliferation and differentiation. Remember, on the humoral side, there's only one effector cell, which are the plasma cells. They're going to produce those antibodies. Those antibodies are going to bind to the target antigen, neutralize it, and target it for destruction. So this T helper cell, your book kind of made it sound kind of weird. It also plays a role in activating the cytotoxic T cells. The only question really is the mechanism. That really isn't quite worked out yet in immunology. It's very difficult to study immunology, and it doesn't always respond the same in a lab as it does in a person. And so T helper cells do have an effect on cytotoxic T cells, even though your book made it sound kind of weird. So once we have activated T helper cells, they're going to do their job. They're going to communicate. The T cell helper cells communicate with naive B cells. They're going to communicate with macrophages and really, really get them all excited through release of cytokines. So then this macrophage, now it's got increased killing power. It's going to go ahead and search out using its toll-like receptors. So looking for those pathogen-associated molecular patterns, the damage-associated molecular patterns, and go look for that antigen or that invader to eliminate it. These cytotoxic T cells, once they've met their antigen, it's been presented, Once they're activated by the chemical signals, those cytokines from the T helper cells, then they're going to go look for that infected cell. So that virus cell or that tumor cell, and it's going to deliver its death package and induce apoptosis, so that programmed cell death. All right, and I just kind of broke this down for you. Populations of lymphocytes. We have this naive lymphocyte, whether it's a B cell or T cell. It just means it's naive. It doesn't know what it doesn't know. It never encountered an antigen before. When it meets the one specific antigen, because once again, each one of these cells is going to be specific for a particular antigen. So it has to meet the one, it has to get the confirmation signal, and then they become activated. And activation, there's two things to it. They're going to proliferate, they're going to clone themselves, and then they're going to differentiate into distinct populations. There's two general populations from that exposure to the antigen activation. You always are going to get effector lymphocytes. These are going to be short-lived. They're involved with that primary response, and their job is to eliminate the invader. So think of these as soldiers. They're going to go into battle. They're going to fight right now. The other population, and this is both on the humoral line and on the cell-mediated side, we're going to get memory lymphocytes. And so these are going to hang back. They are not involved in this fight. So they're going to fight next exposure, so secondary response. These cells are long-lived. They can live for decades. That when they do get activated, the next time the person comes in contact with that antigen, they're going to have a more quick and a stronger response that will bring about immunity, so that person shouldn't get sick. So that's going to be that secondary response. So primary response, definition, first adaptive response to a particular antigen. Oh, and I thought I gave you a definition of secondary response. It's coming up. So secondary response would be any subsequent exposure. All right, so let's look at humoral immunity. So we said this has to do with fluid, so it's going to be those activated B cells are going to differentiate into plasma cells in response to an extracellular antigen, so in the bloodstream or the tissue fluids or on a surface of a mucosa. They are going to produce these Y-shaped proteins called antibodies. And like I said, this is just a nice picture of what antibodies do. Like I said, they don't kill on their own. They're basically going to take that antigen, put it in a big bear hug, and then yell and call the other parts of the innate immune system to destroy it. And so these two parts, so you've got this variable region. So the hands, I'm going to call them the hands. So they say these are arms. So at the very end of arms or hands, that's the specific part. That's where the antigen binding site is. And each plasma cell is only going to make one kind of antibody specific for one antigen. This constant region, I kind of liked your book. They said, oh, these are like little flags, and that's true. So the stem region is going to be constant, and it's constant so the immune system can recognize that it's like a little red flag that's going to mark that bound antigen for elimination. And so we talked about opsonization, and so that just helps elimination through phagocytosis. Some of these antibodies can activate complement, so then we're going to get increased inflammation, increased opsonization, and then we can have the membrane attack complex. That's going to get rid of it. And then we haven't talked about it, but we will. Antibodies can also activate natural killer cells. So now let's zero in on a B cell receptor. So a B cell is going to basically have an antibody embedded into its cytoplasmic membrane. And it's going to have, remember the hands are the specific part, so that's the variable region, that is specific in shape, molecular construct for a particular antigen. And so this is going to be the receptor for that antigen. So these receptors, that's how they're going to recognize their antigen, are going to be on naive cells and also on memory cells. And remember, it's those memory cells that are going to respond really quickly if they meet their antigen again. So now let's look at cell-mediated immunity. Once again, we have those two subsets. On the surface of those cells are markers called cell determinants or cluster determinants. And so, because, hey, all lymphocytes look alike, right? Can't tell them apart. So, I know in lab, I had antibody tags to these. That's how I could sort out my T helper cells from my cytotoxic T cells, because I did work specifically on cytotoxic T cells. So their CD8 cells are those cytotoxic T cells. Remember, their job is to destroy corrupt host cells, so virus-infected cells or tumor cells. Those helper T cells have that CD4 surface protein. And remember, the CD4's job, those T helper cells, they're going to help. They're going to activate, direct, assist. They don't do any fighting on their own, but they do activate, direct, and assist both the humoral and cell-mediated adaptive response. And so this is just looking at a picture, and yes, I told you you'd see this picture again. So here's the CD marker. So it would be a CD4 on a T helper cell or a CD8 on a cytotoxic T cell. This other receptor is just called a T cell receptor, or TCR. It does not recognize free antigen. So this would be the variable region that's going to be specific for an antigen. So they're always showing you with shapes that match. Thing about T cells is they cannot recognize a free antigen. So they will never recognize just like a free virion particle or free bacteria. They can't do that. They can only recognize an endogenous antigen. And so that antigen has to be presented on something called an MHC, which we're going to look at later. But it needs special presentation. So it's going to have to be presented with its antigen by an APC, an antigen presenting cell. So a dendritic cell, a macrophage, or a B cell. Same thing, the helper T cells and cytotoxic T cells must be activated before they can multiply. So there's going to be this little communication between the antigen presenter and that T cell, and then we're going to get some activation because that T helper cell is going to do a whole lot of talking with a whole lot of cytokines, but that's what we need, both presentation of antigen and activation to get our effector population, so our effector T helper cells and our effector CD8 cytotoxic T cells. All right, I know that was a whole lot of talking, but I think this should help.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Okay, so that's a great one to go back and watch when we're all done. 'Cause
Activation.
So that's a great one to go back and watch after we're done with this. You can watch that one a couple of times, actually, because it's just a summation of everything we're talking about. I do want to point out that even though it just talks about B cells and their memory cells, T cells also generate memory cells. The reason we talk about B cells more, it's just easier to show you, it's easier to measure, and it's really hard to quantitate in a lab. So just kind of walk through one more time, and then we're going to move on. So we have naive T lymphocytes. That means they've never encountered an antigen. They have to meet that one specific antigen. For T cells, this has to be presented on an antigen-presenting cell. B cells have those antibodies, so those B cell receptors, they can meet their antigen on their own, but B cells and T cells require a conformational signal. And this is an immune safeguard to make sure that the immune system doesn't have an overactive response or an autoimmune response. So there's a conformation signal. That T helper cell is really important in this conformation signal. It's going to provide that activation for B cells, and it's also required for activation of cytotoxic T cells. Then we have activated lymphocytes. The first thing they're going to do is they're going to proliferate. That just means they're going to divide. They're going to clone themselves, and then they're going to differentiate into distinct populations. And we said there's effector cells, and then there's memory cells. The effector cell for B cells is kind of easier. It's plasma cells, but we're just going to look at T cells. There were the cytotoxic T cells that actually do the killing. They're going to kill viral infected cells or cancer cells through cell-to-cell contact, through apoptosis, nice and quiet. That's that quiet suicide. Helper T cells that can produce cytokines, direct, support, activate other cells, both B cells, T cells, and they're going to talk to the macrophages. They're just talkers. They're communicators. They don't do any killing on their own. You get those macrophages super excited. There's another population we didn't talk about and that wasn't in the video because we don't know a whole lot about them. These are going to be the regulatory T cells. And these regulatory T cells, actually, just what the name implies, it's going to regulate the immune response. So it can stop the immune system from overreacting and responding to harmless substances. So they are sort of a subset of the helper T cells, but instead of stimulating the immune response, they're actually going to dampen it and prevent the immune system from overreacting to harmless substances. And so it's a really hot area of research, especially with the increase in autoimmune diseases. It's when the body reacts to self. I think we kind of covered that, so that picture is there for you if you want to look at it again. All right, and I've also just given you some definitions. So naive, activated, effector, memory, everything I just kind of said, but there it is in word form, because I realize I'm a picture person, but not everyone else is. All right, so our last topic on this section, we have to talk about the lymphatic system. Hopefully you had this in AP2. If you haven't had AP2 yet, here is a little summation of the lymphatic system.
In a nutshell, the lymphatic system is a drainage system that removes excess fluid from body tissues and returns it to the bloodstream. It's actually a subsystem of both the circulatory and immune system. The major purpose of the circulatory system is to bring oxygen and nutrients to body tissues and remove wastes. This exchange happens in the smallest blood vessels called the capillaries. Blood plasma containing nutrients moves out of capillaries at the arterial end of capillary beds, while tissue fluid containing wastes reabsorbs back in at the venous end. However, not all of the fluid is drawn back to the bloodstream at this point. About 15% of it is left in the tissues and would cause swelling if accumulated. This is where the lymphatic system comes into play. It picks up the excess fluid and returns it to the circulatory system. Unlike the blood circulatory system, which is a closed loop, the lymphatic system is a one-direction, open-ended network of vessels. Lymphatic vessels begin as lymphatic capillaries made of overlapping endothelial cells. The overlapping flaps function as a one-way valve. When fluid accumulates in the tissue, interstitial pressure increases, pushing the flaps inward, opening the gaps between cells, allowing fluid to flow in. As pressure inside the capillary increases, the endothelial cells are pressed outward, closing the gaps, thus preventing backflow. Unlike blood capillaries, the gaps in lymphatic capillaries are so large that they allow bacteria, immune cells such as macrophages, and other large particles to enter. This makes the lymphatic system a useful way for large particles to reach the bloodstream. It is used, for example, for dietary fat absorption in the intestine. Once inside lymphatic vessels, the recovered fluid is called lymph. Lymph flow is enabled by the same forces that facilitate blood flow in the veins. It goes from lymphatic capillaries to larger and larger lymphatic vessels and eventually drains into the bloodstream via the subclavian veins. On the way, it passes through a number of lymph nodes, which serve as filters, cleansing the fluid before it reaches the bloodstream. Lymph nodes are small bean-shaped structures scattered throughout the lymphatic network. They are most prominent in the areas where the vessels converge. Lymph nodes contain macrophages and dendritic cells that directly swallow up any pathogens, such as bacteria or viruses that may have been taken up from an infected tissue. They also contain lymphocytes, T cells and B cells, which are involved in adaptive immune response, a process that produces activated lymphocytes and antibodies specific to the invading pathogen. These are then carried by the lymph to the bloodstream to be distributed wherever they are needed. The lymphatic system also includes lymphoid organs. Primary lymphoid organs, the thymus and bone marrow, are the sites of lymphocyte production, maturation, and selection. Selection is the process in which lymphocytes learn to distinguish between self and non-self, so they can recognize and destroy pathogens without attacking the body's own cells. Mature lymphocytes then leave the primary for the secondary lymphoid organs, the lymph nodes, spleen, and lymphoid nodules, where they encounter pathogens and become activated.
Okay, so that was an awesome summation of the lymphatic system. So our goals for this, we're just going to talk about it as the immune system function. So just kind of so you know the roles of lymphatic vessels, the secondary and the primary lymphoid organs in relation to the immune system. So the immune system functions the lymphatic system when it brings populations of lymphocytes into contact with their antigens. Is important because each lymphocyte is specific to only one or just a few antigens. Lymphocytes have to encounter the appropriate antigen in order for there to be an immune response. And so just showing you, we've got all this green stuff is the lymphatic system, and so we're going to look at different parts of that. So first we're going to talk about those lymphatic vessels. And so it's just showing you the lymphatic vessels run alongside the cardiovascular system, so blood vessels. They carry lymph. And so lymph is that excess tissue fluid that enters those lymphatic vessels. And these doorways and these capillaries open up super big. So we're going to get, yes, excess tissue fluid, antigens from tissue, so bacteria, virions, cancer cells, circulating lymphocytes, and other white blood cells. So this is the way white blood cells can patrol the whole body because they can be in the bloodstream, the tissue fluid, lymphatic vessels, and then be returned to the bloodstream and then do it all over again. So big thing, lymph contains antigens from tissues. Lymph flows through lymph nodes. So these lymphatic vessels, you have hundreds, like 600 lymph nodes scattered throughout your body. And so these lymph nodes are going to act like little filters, and there's part of that secondary lymphoid tissue. But basically they're going to filter this lymph in hopes that there was going to be some contact between antigen and lymphocyte. Inflammation, we talked about that as part of that innate immune system, causes these capillaries to get leaky and excess tissue fluid, so swelling. Remember the cardinal signs: redness, heat, pain, swelling. And so we get excess tissue here. And the idea is, yes, we're going to get more white blood cells there, but then we're also going to have excess tissue because we want to force any invading pathogens into the lymphatic system. So as it goes through the lymphatic vessels, it's going to encounter lymph nodes, and it's going to activate the immune system to remove the invader. So the secondary lymphoid organs. So these are sites where lymphocytes are going to gather to contact their antigen. And so I thought it was kind of cute. Your book compared it to a coffee shop. So yeah, Central Perk from Friends. So secondary lymphoid organs. So spleen, tonsils, lymph nodes, appendix. Also something called mucosa-associated lymphatic tissue, MALT, and something called SALT, skin-associated lymphoid tissue. And think about it, you are a house to be protected, so you would always put sentries by your doors and windows, so just underneath the skin and underneath the mucosa, because these are going to be areas of entry. So in the intestines, there's special things called Pears patches in the small intestine. And so these Pears patches are lymphoid follicles. Basically, they're just filled with T cells and B cells. And so what happens is Pears patches sort of just allow the sampling of anything in the intestinal lumen because the food we eat is not sterile. And so via these specialized M cells and dendritic cells, they just sort of get this little sampling of anything in the lumen. And the idea is to prevent that microbial invasion. In this case, Pears patches are going to be in the digestive tract, but we also have mucosa-associated lymphatic tissue with the respiratory tract, the genitourinary tract, and like I said, you have the same kind of tissue underneath skin. But these are going to be places where the antigen and antibody are going to meet. So we have lymphatic vessels are going to carry lymph, and then there are lymph nodes scattered throughout those lymphatic vessels. Secondary lymphoid tissue, that is going to provide sites for the antigen to meet their lymphocyte that is specific for them. So those are secondary, so spleen, lymph nodes, tonsils, Pears patches, that makes up MALT, mucosa-associated lymphoid tissue, and we have SALT, skin-associated lymphoid tissue. And then the last part we're going to talk about are primary lymphoid organs. And so these are where lymphocytes originate and mature. And so number one is going to be the bone marrow, because we learned in last chapter that's where the hematopoietic stem cells reside. So that's going to be the production of all blood cells, not just lymphocytes, but we're just talking about the lymphocytes today. B cells, they not only come from the bone marrow, but they're also going to mature in the bone marrow. And there's something that occurs for B cells, something called negative selection. And so the selective process is part of that maturation. We need to make sure that B cells are not going to be self-reactive, so they cannot bind to self-antigens. And so they go through this sort of selection process, and if they do bind to self-antigens in the bone marrow, then no, you are not good enough. You have to go through apoptosis, you're out of here. And so this sort of negative selection is critical for preventing immune system from attacking the body, so from autoimmunity. So B cells, think B and B. B cells, they are made in the bone marrow, they mature in the bone marrow, and then they're going to be released, and then they're going to go hang out in secondary lymphoid organs, waiting for the day that they meet that special one antigen. So the thymus, this is a butterfly-shaped gland. It is going to be in your mediastinum, so just right above your heart. This is where T cells migrate to. So T cells, so you get T and T. So T for T cells, T for thymus. T cells, they are made in the bone marrow. They're released as immature thymocytes, which are then going to be transported through the blood to the thymus. The thymus, think about it like a school for T cells where they're going to mature and they're going to get educated. They too go through a screening process, a little more brutal for T cells. Only about 5% are going to make them out of the thymus. One, you think about it, T cells have the ability to kill your cells gone bad. So there's this positive selection. First, the T cells have to recognize self, and that's through what's called the major histocompatibility complex. And so think of that like your barcode. You have special markers that say that, hey, this cell belongs to Jim, or this cell belongs to Susie, or whatever name you want to plug in. Think of your name. So if they cannot recognize your self-marker, then they're going to be eliminated. So they're going to go through apoptosis. So that was positive selection. Negative selection, these T cells, also we don't want them to recognize and get all excited about self-antigens, so normal body components, so your normal proteins that are present on the surface of your healthy body cells. And so if they do, just like the B cells went through negative selection, if they bind, then they're going to go through apoptosis. Like I said, this process is so stringent that over 95% of those immature T cells that enter the thymus, they never leave. They undergo apoptosis. Imagine if you fail the test, like, hey, you're out of here. And that's kind of what happens in the thymus. So think T cells made in the bone marrow, mature in the thymus, and then they're released as cute little naive B cells. And then they're going to go circulate throughout the blood to lymph, and then they're going to seed secondary lymphoid organs. So once mature, lymphocytes, both B's and T's, gather in secondary lymphoid organs to wait to encounter their one perfect antigen that they were just destined to meet. Because the adaptive immune response is specific. Okay, we're going to stop here. So this is going to wrap up part 1 of the adaptive immune system. You have an awesome day.